And good morning, everyone. We are ready to go for our fourth day of our summer collaboration today. Just a brief reminder for the students, make sure that you have your introductory slide ready for tomorrow. Again, you can see all the details that you should include on that. This way we get to meet everybody. We get to know who's who in our group. We also have a couple of new teachers who will introduce themselves tomorrow as well. So not only are we going to meet our new students, but we'll also meet some of our new teachers that are new to the collaboration. So let me switch to my slide of what we're doing today. So today, we're going to have two things going on. We're going to learn about what we do with the X-ray diffraction data once we have it. And then we're going to talk about the protein data bank, and Mrs. Mackey is going to introduce us to the PDB and how that works. And then after that, we'll have any questions that anyone has, and then we'll be ready to go for Friday. So let me switch this up. All right. So yesterday, we talked about the light source and how we collect data, right? So we know we have to prepare proteins into crystals. Once we have those crystals, we have to freeze them so that we can preserve them. And we need to preserve them so that they're stable enough so that when we put them in the X-ray beam and collect data, the X-ray doesn't damage the crystals, and so that we can also maintain the integrity of the crystals and the structures of the proteins. But once we have that X-ray diffraction data, the question now is, well, what do we do? Well, we take those spots and we turn them into protein models. And the way that happens is a little tricky. We have to use several programs that do this. So just a quick recap, we collect the X-ray diffraction data at the beam. So right here is the detector, okay? And that detector, which is several $100,000, picks up the X-ray that is coming in through from the beam line. So the X-rays are moving in this direction, and they'll hit this detector, and the detector will pick up that diffraction pattern. That diffraction pattern will then be converted into actual model data. So the first thing we have to do is we have to do a screen or a raster data collection to see if the crystal actually diffracts. So very often we'll have a great looking crystal, we'll expose it to the X-rays, and we may not get the most ideal pattern. Sometimes we don't get anything. If you notice, there's this dark circle here. That dark circle represents ice. So a lot of times we'll get this big dark ring and nothing else, and we just have an ice ring. But really what we want to see is a pattern that looks like what we have here in this particular image. Those little black dots, you can see that it starts up with this circle here, and then there's another circle on the outside and another one. And all these concentric circles of dots represent a pattern that indicates good diffraction. So during data collection, we're basically taking hundreds of pictures of the protein in different orientations as we turn it through the X-ray beam. And as we rotate that crystal, we're going to get a complete picture of what the protein looks like. So once we get that diffraction data, and again, we get that data by collecting data from the hotspots. So this is a crystal with a grid placed over it for what's known as a raster collection. And each of these little points of green indicates where there might be some good diffraction data. Okay. Once we collect that data, we then need to go from the dots to the model. And to generate a model, there are several steps that we have to go through using a bunch of computer programs. And so I have some of the images of the programs here with their logos. We have Phoenix, CCP4, which is actually a suite of programs. And then we have COOP. And that allows us to convert this dot matrix of X-ray diffraction into a three-dimensional model, okay? And so when we look at crystallization data, there are two main types of files that we're going to deal with. There are others that are out there that are processed at the beamline, but pretty much what we get are two files that are output through programs at the beamline. The two programs are called FastDP and Dimple. And what they do is they process the X-ray diffraction data into two files. The first one is a PDB file, and that PDB file contains the structural information about our protein. It includes the number of amino acid residues. It actually outlines the individual residues that are there. It gives us a number on the resolution. How good is our diffraction? How good is our protein? There's another measure called the Wilson B factor and R factor. So the PDB file contains a lot of important information about the structure of the protein. The other file that we use is a file known as an MTZ file, and this is known as the electron density data file. This is our map of our actual structure. So this gives us the map of electrons that are the actual structure, the physical structure of our protein. And so what we need to do is analyze that MTZ file and create a protein structure based on that density. So the MTZ file is really the map. That's the roadmap that says this is what your protein looks like. And then we use that to construct the actual physical structure of the protein. So when we see an MTZ file, it's just basically a blue cloud. And a PDB file is really nothing more than a set of data. But there's some important data that's in here. One of them is resolution, okay? And that resolution number, which you can see here, in this case, is 2. The lower the number, the better the resolution. Now, ideally, when we are looking at proteins, we want to get resolution below 2, all right? And that 2 is representative of 2 angstroms. So anything lower than that number is considered a good structure, because that means that we're able to resolve the differences in the bonds and the individual atoms within that protein. The other number we look at is the R value. And the R value is a measure of the comparison between the model and what we actually have. And the lower that number, the better the refinement. So generally, we try to pinpoint an R factor at or below 0.25 as a starting point. And ideally, if we can get that below 0.2, once we're done refining, we know we've done a really good job on our protein. The other one is the B value. And the B value is a temperature factor. It basically measures how much molecular movement you have. The more things are moving around, the faster things move around, the more blurry they get. So the lower the number, that means that there's less movement, which means there's less blur. So for all of these factors, the resolution, the R factor, and the B factor, the lower the number, the better the refinement, the better the resolution, the better the structure is likely to be, okay? And so this PDB file contains all that information, as well as all the actual details about each residue that makes up the amino acid primary structure. CCP4 is a program, it's a suite of programs that actually processes the data, and that's what produces the electron density maps. So these programs are loaded up at the beamline, and what these basically do is convert that X-ray diffraction dot into that map. And so there are all sorts of programs that are going on behind the scenes that we don't see. There's reduction, there's scaling, there's phasing. And basically what you end up with is the ability to take that data and automatically process it. So automation is what's happening at the beamlines. The beamlines are highly automated. So each beamline takes the data, processes it, which saves us time and effort. And there are several programs that do this. One of the more common ones is called FastDP, which will turn those dots into a FastDP MTZ file. And that MTZ file is the electron density map that we want, which is going to tell us what our protein actually looks like, okay? And this is what that map file looks like, okay? So you can see you have these blue clouds here, which represent our electron density. And this basically shows us where the electrons are that form our protein structure. All of these little lobes indicate places where there might be a nitrogen or a hydrogen or a carboxyl group that forms an amino acid. But this is the actual structure that we have. And so we use this as the map to build our protein, okay? So these programs, DIMPLE, FastDP, Phoenix, and Coot, allow us to superimpose a model in yellow onto this map. And the map is what we can utilize to construct our protein. Okay, so DIMPLE is an automated program that matches the map to the model, and most often this is going to be used to find if there's anything bound inside the protein itself. But it can also be used for related structures, and by related I mean a model that is similar to our protein that's already in the protein data bank is fitted to the data map. And we use that to build a new model. So we know that AMX and FMX can both run this at the beamline. There's a little checkbox that we always check. And one of the things we always say is make sure FastDP and DIMPLE are checked, because that will allow the beamline to actually run the refinement and match the model and the map so that you can actually have something to start with, okay? We can also do this afterwards by CCP4, but it's a little more problematic that way. So usually we like it if the beamline does it, because it will take the data and basically do this. It will fit the yellow into the blue, okay? And then our model and our map are superimposed on each other, and we can start making refinements. Okay. The DIMPLE output makes that map in a model that can be refined and explored in a couple of programs. One of the most common ones we use is Coot. And again, what it does is it overlays the model, which is, in this case, it's green, on top of the electron density map. And then what we do is try to fit these individual amino acids into the cloud. Some of them may be sticking out of the cloud. Some of them might not be near the cloud at all. And so our job in Coot will be to take these individual parts of the amino acids and one by one systematically fit them into that blue cloud. Okay. Once it fits the blue cloud, we now have a structure of our actual protein by using this green, sometimes it's yellow, this green backbone as the foundation for creating the structure. Okay. Phoenix. Now, this is for our computer savvy people. It's a Python. I never knew this is what it stood for. It's Python-based hierarchical environment for integrated crystallography. And they use the X for crystallography. So basically, this is an automated program. What this will do is it will take the data and it'll take our model and it'll automatically generate the electron density map and automatically align the model over the map. So what this will do is use its own computer algorithm to create the most ideal structure based upon the map that we have. It is a very accurate program, but any scientist will tell you that the human eye has the ability to decipher things that automation doesn't always determine. Because with automation, you're basically set to a specific algorithm and it looks for that. But any slight variances, it's up to us to do that. So what we usually wind up doing is taking Phoenix, running that first, and then we use Coot to manually adjust the protein. So Phoenix gives you an output that looks like this. And basically what Phoenix does is it builds the model into the map. So it creates this three-dimensional structure for us. And it also gives a tremendous amount of data. Right here, you can see there are some R values here that it gives. And it shows you also different resolution levels. And what it basically does is it allows you to determine how well the model has been constructed and how well it fits the map. And so it'll give you some output like the R factors, which tell you how accurate your model fits the map. And you can see that, in the beginning, it starts off high and then it drops off low. And you want to see that because that tells you that as Phoenix is running the program, it's finding ways to fit that yellow backbone of the amino acids into the electron cloud. Okay. But it doesn't do it 100%. So we need to move on to another program after that. So Phoenix is initially what we use to set up the model. And then we use Coot to manually refine the model to fit the data map. And Coot is where it gets to be a little more tricky because that's where we individually manipulate each amino acid in the polypeptide chain. Okay. So what Coot does is it provides the map as the basis to generate the structure. And we fit that model onto the map. And that model is, again, a model that we got from the PDB, and it matches as closely as possible to the protein. So each amino acid is individually manipulated to fit the data map. This includes literally not just looking at the amino acid, but the individual atoms, where you will actually try to move carbons and nitrogens and hydrogens around and sections of it around to fit that data map. The ultimate goal is to get the model to fit the map as precisely as possible. So I always go with this idea in my head that the model must fit the map, and how well the model fits determines the accuracy of our structure. So Coot is where we will spend a lot of time individually manipulating these. Usually we'll have multiple data sets. So in class, what may happen is students may work in groups or in pairs, and two people may work on one data file, two people may work on another. And the goal is to try to see which model fits the map the best and with as much precision as possible. Now, once you run Coot, you usually go back to Phoenix and do another automated refinement, and then you go back to Coot and try to finesse it. So basically, when we fit the structure to the data map, we keep doing this in cycles where we go back and forth. And again, the yellow is the model that we identified as the close match. And again, the way we find that model is searching the PDB for a protein that is most closely related. The more that this model fits the map, the more accurate our protein is going to be. So if you look right here, you can kind of see where this double ring structure is here. You see how it sits nicely inside of this cloud? Okay, that's what you want. You want your protein structures to fit nicely in the cloud. Same thing over here. You can see right here in this area that everything is nice and centered, as closely centered to the cloud as possible. That's a good model fit. Okay. Once we do that, and we get that match, we'll use all those different commands to try to twist and turn and go back and forth, trying to make sure that this backbone and all of these little components that stick off of it, including side chains, fit as well as they possibly can. Okay. Now, Coot also lets us validate the model. So after we refine it, it will analyze the refinements and tell us, do your refinements make sense? You know, visually it fits, but how well chemically do they fit? And so there are different analytics that we can use that tell us whether or not the chains are precisely where they're supposed to be. There's another one which is known as a density fit analysis, and this tells you how well they fit. And the areas that come up red, those are hot areas, which are indicative of, hey, this may not fit perfectly. And then you can go right onto those, click on them, and it takes you right to the residue. And then you can manipulate the residue further and try to get it to fit the cloud. And again, the whole idea is get the model to fit the map. Make sure the model and the map are as close as they possibly can be. Okay. And again, you do this through several cycles. So it's kind of circular, right? You do Phoenix, then you run Coot, back to Phoenix, back to Coot, back to Phoenix, back to Coot. And you keep doing this until you get to the point where your model really can't be refined any further. Okay. Then you're ready to deposit. And the whole point of getting ready to deposit is when you see that your refinement data really doesn't change much. Okay. Your R values are not getting any lower. Now, sometimes they'll actually go up a bit. So what'll happen is you'll refine a file, run it through all these processes, and then when you start to refine it again, you'll notice that, hey, my R values are higher than they were the last time. That means that you may have actually over-refined it. So that means that you've done as much as you can do, and you need to go back to the original file so that you can actually use that. The other thing we look at is the validation of the model in Coot. If it shows a very good fit, we've done a good job. We also want to eliminate what are known as outliers. Some of the amino acids might be just sticking out all crazy and wrong, and no matter what you do, you can't get them to fit. So you actually can delete some portions of that. Once that's all done, we then take this data file and we run it through the protein data bank and run what's called a validation report. And that report will analyze the structure and it will inform us of any problem areas that are picked up. It's sort of a last check that you can see to determine whether or not your structure looks really good. Okay, and that report is really easy to read. Here's an example of one. It will give you the simple graphical output and basically tell you some simple things. Number one, it will tell you your resolution, and in this case, it's 2.92 angstroms. Now, if you remember from the previous slide, a couple of slides back, what's the minimum resolution that we want to be at? Or what's the threshold for that? It's 2. This is 2.92. It's a little high. Okay. Then when we look at this overall quality at a glance, there's a percentile score, which allows you to see how well your structure looks compared to all X-ray diffraction structures, as well as structures similar to it. Anything in this red zone, which you see the circle here, means that it's a lot lower than what it should be. So that means that this particular structure is worse than what's in has been deposited to the PDB, and it's worse than other structures. And you can actually see that all the indicators are in red, except for one over here. Okay. So that tells you just at a glance that this probably isn't a really good refinement. All right. The next one gives you some numbers on it. And again, here's our resolution, 2.92. That's pretty high. Our R factors, we want them to be like 0.25. This is 0.479. So this is almost double what it should be. So that tells you that it's probably not that good either. The B factor is also kind of high, which tells you that there's a lot of molecular motion, which means there's a lot of blurriness to this particular protein. So this would probably not be something that could be deposited. Actually, it's definitely something that cannot be deposited because it's a very high resolution. The R factors are very high, which means that the model and the map don't really fit well, and there's a lot of blurriness to it. So what would a good one look like? Like this. You see where almost all the indicators are in the blue range, and even this one here, these RZ outliers, this is kind of in this pink. People really don't worry about this too much, I was told, but these indicators all indicate that we've got a pretty good structure. And look at our resolution, 1.75. Okay, so that's considered a much better structure, and this would be something that would qualify for being deposited. Okay, and then if we look at other factors, look at our R factors, 0.21, 0.20. Look at our B factor; it's down to 31. So that tells us that we have a very good structure. Our map and model match really well. Our resolution is excellent. We can really decipher between different atoms and see lots of differences between different atoms. And our B factor is low, which means we don't have a lot of blurriness going on there. Okay. Another one is the Wilson B factor, which is 26.5, very low. So that means we've got a pretty good structure. And this is actually a structure that was deposited to the PDB. So this is this one, which is this TRMD, known as 8FCI, which was deposited back in December of 2023. So once you have all those factors figured out with your structures and you have a good report on your validation, then as a group, the team can deposit it. And so these are the students that I had involved with this and some of the teachers. And then here are our laboratory people that help us out. And you can see right here, here's the diffraction data. Okay. And again, a good resolution, a really good resolution actually. And pretty good R values. They're below 0.25, so we have some good numbers there. Okay. I want to take you on a quick peek at what COOT actually looks like. So I'm going to open up COOT so that you can see it. And
can I interrupt for a second?
Yeah, go ahead, interrupt. Yep.
So because one of my functions in this group is to translate for the people who have never been exposed to this information before.
Yep, go for it.
Because Mr. Bolin like lives in this pool, and I only get to work with the program occasionally. And thank you, because you cleared up a lot of things about, especially like the B factors and the R factors. I always knew we were aiming for low numbers, but now I know why.
You know why, right?
Right. But for everybody else, when Mr. Bolin showed the picture of that blue protein, of that blue cloud, and then that's the map, and then the structure, which were the yellow and green like ball and stick things. I want to make sure that everybody understands what we're talking about. We know the amino acid sequence of every protein. If there's a similar protein already known and its structure is already known, you can pull that structure out of this PDB and make minor adjustments based on how yours is different. And that's what generates that green or yellow ball and stick thing. The blue thing is what's generated at the beam line. So it's a snapshot of, here's the way I like to think of it. It's a snapshot of the shadow on the wall that the protein made while it was moving. And you have the hands. Let's pretend it's a puppet figure. You're making shadow puppets. The fingers, your hands and fingers are the yellow or green stick model, the structure. Your job is to take the structure, the fingers, and make sure that everything fits inside the shadow. That's the basis of what he's doing. So this would be the shadow on the wall. And then when you add in what's known about, we know these amino acids are what make up the thing. You got to make sure that because a skeleton can be in any position you want it to be. You got to make sure that you get that skeleton of the protein into the shadow that was created. And Judith has raised her hand.
Yes, I have. Quick question. So you kind of answered part of it, which is that you do know the amino acid structure or the amino acid sequence. So when you're taking the model, and let's say you find something that's similar, but a couple of the amino acids have to get changed out, does the program do that for you, or is that something you manually have to do, is change out one amino acid for another? I mean, I understand that you're looking for IMF connections that are common within that structure that would help you generate folds and such and get a start on the mapping process. But which one would actually exchange out and make sure that the model actually includes all the same amino acids as your protein?
Yep, you would manually remove them. So generally,
so the program does that for you?
No. So
that has to be done before you create that initial PDB file.
Right.
Or the initial PDB file comes, and then you can change the residues, the amino acid sequence in it.
Okay, so you do that first, then that runs into Phoenix?
Mhm.
Okay.
Or you could also do it after you've run it through Phoenix. So you can go back in and you can change the sequence afterwards, because I think you can change it before it goes into Phoenix. I'm always changing it in Coot. So once you get your model and your data, the first thing I always have the students do is I give them a printout that shows the amino acid sequence, and I say to them, here are the highlighted 15 residues you have to go in and change. Change those first. And you go in and you simply do it's a real simple process. It's just, it takes time. And you can mutate. It's called simple mutate. You can mutate one amino acid for another. Once that's done, then you go in and you start manually adjusting it.
Okay, and that's in Coot that we do that.
In Coot, correct. Yep.
Okay. And then we'll have a model, and we can still use known structures from previous, known connections and map them in for starters. Okay.
Right. Yep. Technically, it's called molecular replacement, because what you're doing is you're taking the original molecule and then you're replacing the individual residues. Then that becomes the actual new structure, right? And then from there, you start refining it.
Thank you.
Got that? Does that work? All right. All right, Kelly, you're on. Can you see my screen? Is my screen up with the blue cloud?
Yes, I see the screen.
So what we're looking at here, we're in the Coot program. So this is the actual electron density that is produced by the beamline, okay? And so when you look at this, if you're looking at it for the first time, it looks like a bunch of blobs. But if you've done this for a while, you can kind of pick out that there are areas that it looks like there's like a little loop over here, and then there are some lobes. So you can actually then superimpose your model onto this. So the model is what we got from the PDB. Ooh, let me just pull my display manager up. And the model fits like that, okay? And if we zoom in, we can see that here's the first amino acid. Now, in this particular case, is number 16, right? But as we move along, you can see this chain here fits within this cloud pretty nicely, okay? And so if we move along the cloud, we can see where each amino acid fits. We can see there's a side chain here. This is tryptophan. It's my favorite amino acid. Yep, I do have a favorite amino acid. And you can see that it's in that cloud. What Coot allows you to do is rotate this so that you're almost in 3D, and you can see from all angles how that fits. Now, if you wanted to adjust this, there are ways to do this in Coot. One of the simplest ones is this little, it's called real space refinement. And if you notice, I click on this and it moves it and tries to fit it so that it fits precisely the way it should. And then on the side of the screen here, can you see that with the green squares? Is that on there?
Yep.
Okay. That tells you that, oh, that fits really good. And so you can accept that. And this is literally what we will do to manually refine. You go amino acid by amino acid and you look to see, how does this fit? Can I make this fit a little bit better? And the easiest way to do it is by real space refine. Now, let's suppose that one of these you say, I think I could get this to fit better, right? So when you're in real space refine, you click on the residue, you can actually drag the residue and move it around however you want. And if it doesn't fit, you can see what happens. I get all these red rectangles here, and the computer program's really not happy with this to the point where it might actually crash. And what it'll do is it'll try to move it into a different conformation that works. So this is a conformation that works, but it doesn't necessarily fit that. So we can just reject that and put it back in. Okay, but that's basically what we do is you click along, and as you move through, you try to see areas where it fits the density really well. And so far, this one fits pretty good. But there are areas where, like here, ooh, this is a little messy. You see how this is kind of open? So then what we do as a scientist is determine how much of this do we want to keep. You can delete sections. You can actually delete a whole region if there isn't any density surrounding it. But the idea is you want to make sure that your structure fits this cloud as precisely as possible. And areas where it doesn't fit, you may need to cut off a section or cut off an atom, like this oxygen, maybe we can cut off, maybe this one we can cut off, so that the whole thing fits the structure as well as you can. And you can see that this here is pretty big. This is a dimer of beta lactamase. So there's one here and there's another one over here. Actually, there are, let's see how many chains. Yeah, there are 2 chains. And each chain has quite a few amino acids in it. So you're talking about 319 amino acids. There's a zinc in there somewhere. So just this one chain here has 319 amino acids. The other one has another 319. So you're talking about over 600 residues that you have to go through individually. And this does take a lot of time, but this is the piece that's most important because you want to be as accurate as you can be. And we have Kevin, our scientist, who helps us out with this constantly. And his role is really to make sure that we're doing this correctly. And once we have a structure refined, we usually send him the files, and then he'll send it back to us and say, okay, here's what you need to do. You need to fix this. You need to tweak this. You need to run it through this program. So he does give us a lot of guidance on what to do before we're ready to deposit it. Okay. There was a question. Go ahead.
Yeah, the section of the blue cloud, that's what we got from the diffraction, from the X-ray crystallography.
Correct.
So how do we extend that to the larger structure so that we can predict what the larger structure is?
So as you click through, it actually takes you along and it'll move you along. So as we're, like I'm at number 30, 31, 32, see how I'm clicking? And it's moving me along the sequence. And what it does is it has a spotlight area. So it only spotlights a certain region. So there is density here. It's just not showing it because it's just giving you like a focal point to look at.
So
But if I would,
when we look at that small area that you first identified where we get good diffraction, and then you said that's the area we're going to see. How large in terms of molecular structure? How large is that region going to be that we're going to actually get a cloud for?
This looks like, to me, it's picking up maybe a dozen residues or so at any given time because you're zoomed in. Usually you zoom in really close. That's why they give you just this small little spotlight. And I guess just it doesn't eat up CPU space. So really, like, you're looking at, maybe a dozen residues here.
Right, but when you get the diffraction pattern, when you run the beamline, how much data do you get? I understand you're only looking at a piece of it. How much data would we actually have?
The whole thing.
You'd have the whole thing and then more around it. You'd have more crystal.
Okay.
Okay.
So like right here, there's another crystal here, right? That's part of this. Remember we talked about the unit cells and we talked about how they're arranged in line with each other. So there would be another one here somewhere, right? But it's just not showing on our model, but there is lots of data all around this. But we're just focusing on the one unit cell or the one crystal that we have. And so that's what we're looking at in this region here. But there's actually data all over the place. But just to make it easier for the eye, Coot just kind of focuses it down just to this one particular region so you can see it. But this is the whole protein. So that's the whole protein there. Actually, it's a dimer. So technically, like the right side here, there's one lobe and here's another. Those are 2 separate monomers that came together.
And you may have picked up in that crystal, you could have picked up 4 or 5 or 6 of these units. You just identified the one of them.
Correct. Right.
Okay.
Yep. Because once you refine it, it's going to be the same all the way through. Like you're just refining that particular unit of the protein. That's what we're looking at. Yep. Okay. And so anyway, so that's what Coot looks like. So I just wanted to give a preview of, what it looks like. It's actually, it's funny. And Kelly says this all the time, Ms. Mackey, that there's an art to crystallography. And this is the art part. This is the part where you start to artistically look at this and see where do these fit? How does this fit the cloud? Where can I manipulate these things? And you can actually find yourself getting lost in this for hours doing this because you're really trying to get everything to fit as precisely as it can be. And you're looking at angles and you're looking at the cloud and you're looking at different atoms. So it really becomes something that is in the eye of the beholder of how well it fits. So no 2 scientists, no 2 students, no 2 teachers will look at this and come up with the same exact thing. Some people have different thresholds for what fits and what doesn't, and this looks good to me or this doesn't look good to me. So there is an art to it, which is kind of a neat thing that even though you have this data, like your data is the data, like the blue cloud's not going to change, but you can make a determination as to how much of a fit this has that looks good to you. And even when you run your validation and everything, there is a certain range of error, and then everybody has their own preference to what they feel is good enough to use or not. So it's really, there is an art to it, which I kind of like. So it's not just pure science. There's a lot of variability in there too, in a good way. Okay. So I'm going to stop sharing there. Does anybody else have any other questions, students or other teachers? All right. So we're going to spend a lot of time learning how to use these programs. They are kind of complicated in the beginning, but then once you get the hang of it, you really wind up using the same like 6 or 7 commands in Coot. There's really not much more to it. There are other things that you don't really use unless you have major problems. And if you're having major problems, you're probably not going to use the structure anyway, because it's probably a bad data set. So there is a lot of repetitiveness to it. So we will start training with simple models before we start using our actual proteins. And by the time you get to the protein, you'll have a pretty good sense of what to do and how to use it. And it's also an old program, so it runs on pretty much any computer. So that's the good thing.
That's Coot.
Yes,
not
that's
Phoenix.
Coot. Right. No,
Not
Phoenix.
Phoenix.
Not Phoenix, right. So Coot, Phoenix is a much newer program, and you need to make sure you have a good computer for that. And so we will we'll talk about that when we do the trainings and everything. All right, so I'm going to turn it over to Mrs. Mackey so she can talk about the protein data bank and how we use that for our protein structures.
Well, yeah, and I'm just going to say this as the intermediate person, we just, in my opinion, Mr. Boland did a great job of giving you a really more detailed overview than just an overview of Phoenix and Coot and refining. And he saves all these recordings. So if because weeks are going to go by, possibly months before you actually start working with the program, it depends on how your school's thing is, you can always go back to this recording and remember what the Wilson B factor is and the R and basically learn the interaction of, well, where does the dot PDB and the dot MTZ and where do they all fit in? It was a really good overview. I'm planning on revisiting it occasionally. Okay, so I'm going to do this, I think. Am I sharing my screen? What's going on? Share that. And
Not
then
yet.
I can, and then I can do this.
Yep.
And
There it is.
There we go.
Okay.
Okay.
You're on.
I'm on. So hitting my slideshow. You're going to have to show me how to do the disappearing ink thing because I'm not really sure how that happens. But so the protein data bank, the PDB, we've only been talking about it every day so far. We've been name dropping this thing. So it's time to find out what it is. And basically it's a collection of all the known protein structures that have been resolved to a point where the scientific community has accepted them and said, this is a good thing. So there's detailed information about every single protein within that PDB entry. Every protein has its unique 4-character code, the name of the protein, the organism it was isolated from, its amino acid sequence is there. So again, when you get your PDB file, if the protein itself, if you're just looking at a different variant, let's say, of a protein, you're probably going to have an amino acid sequence that doesn't need a lot of tweaking. If the protein you're working with is so different from anything else that's been deposited, you're going to have some work to do. You're going to have to figure out and go in and highlight, replace amino acids as before you can put anything into Phoenix or Coot. The conditions under which the structure was resolved. So this is the plating and all the other stuff, that the metadata we talked about, any other ligands that might be there or any variants that might be associated with it. And then if you're lucky, there's links to the published paper, because what we've been kind of ignoring, the elephant in the room for a long time, is the point of this is to publish. And if you're going to publish your own paper, you need a thorough knowledge of the protein, not just the structure. That's great, but you have to do some background talk about why we're bothering to look at it. Why is it important? What role does it play in disease or metabolism? So having that published paper would help you too. And then the 3D visual molecule, and we've seen that before too. So many things. So all you need to do is Google protein data bank, and this is what comes up. It is open access. You don't have to make an account, but if you know you're going to be working on one particular family of proteins or one group, I like to do it. So you just make your little PDB account up here. And then we dive into the website. I'm going to give you a quick overview so that you know most of the graphics that you're about to see, I stole from one of their training videos. I didn't make them myself. But so the PDB at the top, you have all these tabs. And every single one of these tabs has rich drop-down menus with tons of information. You will hopefully get up to depositing and visualizing, because that's what you do when you add your protein to this scientific discussion. But for now, you're probably going to spend a lot of your time on search, because that's how you're going to get your background information and that model that you have. You got to make your model fit your data, or the structure fit the model. That's where you're going to get your structure from. So at the very top of the page, there's this white search bar, and you can type in, like they say, you can type in the ID, you can type in the author. They explain everything you can do. There's your once you have found it, you may get a whole page, depending on what you've put in. If you've put a protein name into that search bar, you may get a whole page of related or anything that comes up with that search term will show up. So here is 6BE4. It is a protein. It tells you exactly what it is, what the protein is. It gives you what it's classified as, where it works in the I guess, living systems. The organism where it had been expressed from. Most of ours come from bacteria, although here in the protein data bank, they have them from vertebrates, including humans and chickens and all sorts of things. You kind of have to know also, like, the difference between a regular bacteria and an archaebacteria, because that could change your filter searches. And then when it was deposited, so deposit is when you actually submit it. But anybody who's been working online knows that sometimes you submit something and it takes a little while before they agree to make it public. So that's basically it. They deposited this in October of 2017, and it was finally published to the PDB in February of 2018. Then we've got exactly how they got it. You've got the resolution there. All these things that Mr. Boland has gone over, and now because he did them, made my job so much easier. There's that quality score, that sliding red and blue scale, and then here a link to the published article if it exists. And you can keep going down the page. There's so much more. Just about everything on here is clickable and brings you to some other piece of information you need to know. And even then, across the top, forget scrolling down, across the top, there's all of these other things. The experiment would be the exact conditions that were run. The sequence, that's where you'll go to get the PDB file you're going to need to feed in to Phoenix and CCP4. And then the 3D view. Versions would be, this might be particularly useful if, and I'm pretty sure this is the case, the protein you're working with hasn't been really deposited. You're looking for something kind of close to it. That might be where you wind up going. So here's a whole bunch of different versions of 6BE4. Anytime they refine it, they make it to a better resolution, or something has been improved on it, they give you this. The 3D view is, again, all of that stuff that Coot finally did. When you decide you've got that structure and it fits perfectly and you deposit it to this PDB, this is what you get. And there's a bunch of different programs that'll help get this. This is a publishably high quality image of exactly where all of the amino acids are situated in relation to one another in the crystal. And for me and anybody else who needs it, there's plenty of places that you can go to brush up on any holes that you have in your structural protein knowledge base. You can go to Learn, and they can tell you anything you need. So much, and including they've got the molecule of the month. So right now, this is a current screenshot of the molecule of the month and anything you need. So again, depositing and publishing to the PDB. Well, depositing to the PDB and obviously having them accept it and publish it to the real world and the scientific community at large. That's our big goal. And then we would also really love to turn that into a paper, although we're learning that one structure by itself does not a paper make. That the scientific community really wants a bunch of things, but the protein under different conditions, or related proteins all in the same paper, or a protein with a bunch of different ligands associated with it. mutations, or whether you've got different versions of it and mutations. Some of these proteins were resolved a long time ago with low quality. So all you'll be doing is using better equipment and better techniques to get a higher resolution. And we've talked about this. So this is all stuff that we've already done. We keep refining our presentations, and I don't need to say a whole lot. So there's lots of work to be done. So Spark students have already deposited 5. Dr. Pepinella mentioned 8, but the 5 that I have are 6 PEY, 6 UEV, 8 FCH, 8 FCI, and 8 XTA. And obviously, you learn pretty quickly that the 8s were deposited more recently and the 6s further back in time, so that when you see a data bank 4-letter ID that starts with the number 2, you know it's been it was deposited quite a while ago. So that's when those versions will come into place. Has it been updated since then? So I have a link to the PDB in this presentation, and again, I'll post this onto the stream, but all you need to do is a quick search, and if you search for 6 PEI, you'll see that it's got a 2.8 angstrom resolution, which Mr. Boland just told us is not the best, but it was a really novel structure, so that was great. And we have 3 proteins this year. And I don't know, I did a quick search myself, and I can't, we have 7 proteins. We talked about that already, but there are 3 that we're planning on focusing on. The first one, the dihydroorotate homodimetric type, that was isolated from this bacteria. I'm just not going to torture you with my trying to sound it out. Has a similar protein that was isolated from E. coli. So its number is 3MJM, and it is similar to this, which would be a good starting point for us to look. The cyclophilin B, there wasn't a lot of this. I want to say might have been 2. And 1 VAI is a similar one, also isolated from E. coli. And then the third one, the methylmalonic acid, semialdehyde dehydrogenase. This has almost nothing. There were some semialdehyde dehydrogenases. There was 1 methylmalonic acid that popped up, but it looked it had a lot of going on. This is going to take some digging to work with, which could wind up being really interesting for people. But that's the direction we'll be going in with our 3 proteins, and more on those later next week. These are the reference videos if you wanted to basically go through the march through of the PDB, although I find the website pretty intuitive. So once you're there, just start clicking around and you'll find just about anything you want. Does anybody have any questions? I'm going to take that as a no.
All right. Yeah. All right. Thank you. So again, the PDB is a great resource. We can get the sequence files from there too. So the file that has the sequence of amino acids, we can get that from there. And then the sequence of amino acids from our proteins comes directly from Seattle. So we can compare those 2 sequences to each other. There are actually programs that we can use that allow you to do a quick sequence comparison, and it immediately tells you where you have differences in 300 or 600 amino acids. But again, we're going to be utilizing that PDB a lot. It's a really important tool for us, especially when we're trying to figure out different combinations of conditions and different types of experiments and how that's how that happens. So, but we'll be exploring that a lot further. So thanks, Mrs. Mackey. Great job. Any other questions? We're good. All right, so it's 10:52. We'll let everybody log off. Remember to have your slides ready to go tomorrow in the presentations. We're just going to march right through them, and then we'll also have our teachers that are new to the collaboration also introduce themselves as well at that time. So thanks, everyone. Have a great day, and we'll see you tomorrow.
Hey, Rob, I know you have something to do with Sharon, but I have a quick